As we all know, NAVS focuses on the reduction of animals in the labs and therefore gave prizes to projects that match with their outlook. The purpose of an animal experiment is to check whether the same treatment would be applicable to humans. But with an animal model, I had previously read and learned that the time to take care of a mice after injecting a tumor into it was 15 days to 1 month, while in comparison a 3D was at maximum 2 weeks. And furthermore, after the experiment with that particular mice was done, you had to immediately euthanize it. And that is why I asked my mentor if I could do a 3D culture system instead. A 3D culture system has many benefits over not only the animal model, but also other methods. As you can see in this chart over here, the 3D boasts the highest clinical relevance, yet has a lower cost and is more time efficient than the animal model.
In the field of neuroscience, various kinds of animals are used, from small flies like Drosophila to zebrafish to mice and rats, and then also to higher animals such as monkeys. However, in my specific field, on which I was working with the human brain organoids, or let's say the organoids in general, there are not many animals used, since that's one point of organogenesis, that we can study the human tissue, so we don't need an animal which is closely related to us humans, but still it's not exactly the same.
iPSC technology allows generation of disease-specific cell types that are otherwise not easily available for modeling studies. iPSC model that recapitulates human bodily phenotype in vitro cultures is a novel technique and overcomes many of the animal model limitations, so it was more appropriate for my work. With stem cell scientists and human geneticists who explained the advantages of iPSC-based methodologies in understanding human diseases, and this exposure shaped my decision to use this methodology rather than an animal model.
From the very beginning of my research career, I knew that I did not want to rely on animal models, but instead rely on copious amounts of already existing data to build both a humane and scalable process. I was still able to very effectively conduct my research even without the use of animals, and all my teachers and mentors were always very supportive of my research and methods, and they never tried to push me towards using any sort of animal alternatives. It's much easier than expected in most cases to integrate humane methods into research, and integrating these methods will often improve your research because it's a forcing function to develop more scalable methods. It's much easier than expected in most cases to integrate humane methods into research, and integrating these methods will often improve your research because it's a forcing function to develop more scalable methods.